All right, so when you guys have are ready to start today, I'm gonna have you go back to your table 1. Just on this lab part, some of us had a little bit of difficulty struggling with figuring what to do with the numbers here. Okay, so let's say that the total cells here in photo 1 were 60. Oops, that's a gross color. And then total cells here were 120. Okay, when we're comparing, when we're trying to find the average here, okay, if we use these numbers compared to these numbers, so let's say in interphase we had like 50 out of 60 and we had 115 out of 120 going through interphase. If we were to find the average here, okay, we would find that our average would be somewhere, here, let me calculate it really quick, 50 is 107.5. Okay, this standard deviation is going to be massive, okay, when we try to find the standard deviation for interphase there. Okay, so what we need to do is we would need to, what we can do to make our data more accurate so that the standard deviation isn't huge and that we can compare our data between this first set and this first set, because you're each going to be finding a different standard deviation, right? So to make that more comparable, what I'm going to have you guys do is find the percent of cells in interphase and the percent of cells in mitosis, which will be a little bit easier. So here, go ahead and calculate the percent of cells in mitosis and interphase. So for example, like 50 divided by 60 times 100%. You'll have a number there, you'll have a number here, 15 times 120 divided by 100, 115 divided by 120 times 100%. Okay, you'll have a couple numbers here, do that for these two as well, and you should get an average of cells in interphase, which then you can use for your X bar here. And then when you're solving for this, you can just use those percentage, the percent of 50 divided by 60 times 100%, okay? Those individual percents for this set of cells, this set of cells, this set of cells, this set of cells is going to be each of your X values here, okay? So for X, just plug in that percentage for each of these cells compared to the total cells for interphase and then for mitosis, and that should be a more accurate standard deviation for you. I'm going to check those when you guys turn this in, okay? And then I'll give you guys feedback afterwards to see how you did. For this portion of the lab that you're going to be doing today, I'm going to have you read through the background. Pause the video, read through the background, and then answer these 3 questions on your own, okay? Answer those 3 questions. So here we have 2 samples of data, a small sample and a large sample. Comparing the number of cells going through mitosis compared to a large number of cells going through mitosis compared to those that are not treated with lectin, okay? So to determine whether or not lectin influenced mitotic rates of root tips, we'll be performing a chi-squared analysis on the samples to see if it's a statistically significant difference. So what you're going to do first is you've already actually calculated the percent of cells in interphase and mitosis. So go ahead and multiply that by the total cells in tables 2 and 3 to get your expected, your expected values of interphase that you're going to use later in the chi-square table, okay? So you can see here, those values for the expected number of cells in interphase will go here and here, here and here, and then your expected values for mitosis will go here and here, depending on what percentage of cells you think would be in those. And make sure that you're writing them in the right spot. So remember, this is for the small size, this is for the large size. So you're doing 2 different chi-square analyses. Now for your degrees of freedom, I'm going to give you this one because it's in the lab manual, but it's not in this handout. Your degrees of freedom equals the number of treatment groups minus 1 multiplied by the number of phase groups minus 1. So if you look on the last page, which I can't go back to unfortunately, there are 2 treatment groups. There's the control group and the treated with lectin group. So there's 2 treatment groups. and 2 phase groups, which are interphase and mitosis. So our degrees of freedom are going to be 2 minus 1 times 2 minus 1, which equals 1. So for your degrees of freedom, you can have 1 in each of those. And that's what you'll be looking at this row on your probability table. And then make sure you write out your statement, whether or not you reject or accept your null and why. And then to wrap up your lab, we're going to construct a CER explaining the influence of lectin on mitosis. And for the CER, go ahead and write 2 pieces of evidence that support your claim, and try to make this concise. Your claim, evidence, and reasoning. Make it concise. Make sure it's a paragraph, in paragraph form, and try to write this well, but don't overdo it, right? You shouldn't be writing more than, at most, 2 paragraphs, and each paragraph having 4-ish, 4 to 6 sentences. And then number 2, create a pie chart showing the percent of time a cell is in interphase versus mitosis. And then write your brief description there. Answer the 2 questions 3 and 4. And if you have questions as you're going through this lab, please ask. I would be happy to chat with you. Just pop onto the Zoom and we can talk about it. But make sure you try your best first after watching this video. And you should be good. All right. Thanks, guys.